A very warm welcome to Kallang platform Igniting Curious Mind, where you and I grow together as on date 6 June 2026. Let's begin the chapter Anatomy of a Flowering Plant. As you can very easily see the structural similarities and variations in the external morphology of a large living organism, both plants and animals. Similarly, if we were to study this internal structure, one has to find the several similarity as well as differences. This chapter introduces you to the internal structure and the functional organizations of higher plants. Study of internal structure in the plant is called as anatomy. Plants have cells as a basic unit. Cells are organized into tissues, and in turn the tissue is organized into organs. The different organs in the plants have shown are differences in the internal structure. Within the angiosperm, the monocots and the dicots have seen to be anatomically different. The internal structure also shows adaptations to diverse environments. The tissue system. We were to discuss types of tissue based on the types of cells present. Let us now consider how tissues vary depending on their locations in the plant body. Their structures and functions would also be dependent on the locations. On the basis of the structure and locations, there are three types of tissue system. They are epidermal tissue system, the ground or the fundamental tissue system, the vascular or conducting tissue system. Epidermal tissue system is a form so the outermost covering of the whole plant body comprises epidermal cells, stroma, the epidermal appendages, the trichomes and hair. The epidermis is the outermost layers of the primary plant body, and it is made up of elongated compactly arranged cells which forms a continuous layer. Epidermis is usually a single layer. Epidermal cells are parenchymatous with small amounts of cytoplasm lining the cell wall and large vacuoles. The outermost outside of the epidermis is often covered with a waxy thick layer called cuticle, which prevents the loss of water. Cuticle is absent in root. Stomata are structures present in the epidermis of the leaves. Stomata regulates the process of transpiration and gaseous exchange. Each stomata is composed of two bean-shaped structures called as guard cells, which enclose the stomatal pores. In the grasses, the guard cells are dumbbell-shaped. The outer walls of the guard cells always away from the stromal pores are thin, and the inner walls towards the stromal pores are highly thickened. The guard cell possesses chloroplast and regulates the opening and closing stomata. Sometimes a few epidermal cells, which are vicinity of ground cells, become specialized in their shapes and sizes and known as subsidiary cells. The stomatal aperture and the guard cells are surrounded by subsidiary cells which are together form the stomatal aperture. Epidermal cells, subsidiary cells, chloroplast, guard cells, stomatal pores. The diagrammatic representation of stomata with the bean-shaped guard cells and stomata with the dumbbell-shaped guard cells. The cells of the epidermis bear a number of hairs. The root hairs are unicellular elongations on the epidermal cells and helps absorption of water, minerals from the soil. On the stem, the epidermal hairs are called as trichomes. The trichomes shoot systems are usually multicellular. They may be branched or unbranched and soft and stiff, and they may even be secretory. The trichomes helps in preventing water loss due to the transpirations. The ground tissue system. All tissue except epidermis and vascular bundles constitutes ground tissue. It consists of simple tissue, parenchymatous, collenchyma, sclerenchyma. Parenchyma cells are usually present in cortex, pericycle, pith, and medullary rays, and in primary stem and roots. In the leaves, the ground tissue consists of a thin wall chloroplast containing cells and is called as mesophyll. The vascular system consists of a complex tissue, phloem and xylem. Xylem and phloem together constitute vascular bundle. In dicotyledonous stems, the cambium is present between the phloem and the xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissue. Hence, it's called as open vascular bundles. In the monocotyledon, the vascular bundles have no cambium present in them. Since they do not form any secondary tissues, they are referred to as closed. When xylem and phloem within the vascular bundles are arranged in an alternate manner along the different radii, arrangement is called as radials, as in roots and in conjointive vascular bundles, the xylem and the phloems are jointly situated along the same radius of vascular bundles. Such vascular bundles are common in stems and leaves. The conjoined vascular bundles usually have phloem located only on the outer sides of the xylem. For a better understanding of a tissue's organization of roots, stems, leaves, it's convenient to study the transverse sections of a mature zones of these organs. A dicotyledonous roots. Look at the figure. It shows a transverse section of a sunflower. The internal structure, internal tissue organization is as follows. The outermost layer is the epibella. The many of the cells in the epibella protrude in the form of unicellular root hair. The cortex consists of several layers of thin walled parenchyma cells. With the intercellular spaces, the innermost layer of the cortex is called as endodermis, comprises of a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as a radial wall of endodermal cells have a deposit of water impermeable vasc material subbrine in the form of Casparian strips. And next to the endodermis lies a few layers of thick-walled parenchymatous cells called as pericycle. Initiation of lateral roots and vascular cambium due to the secondary growth takes place in cells. The pith is small and inconspicuous. The parenchymatous cells, which lie between the xylem and the phloems, are the conjunctive tissue. They are usually 2 or 4 xylem and phloem patches. Many cambiums ring develops between the xylem and phloem. All these tissues are inner side of the endodermis, such as pericycle, vascular bundles, pith, constitutes the steels. The monocotyledonous roots. An anatomy of a monocot roots is similar to the dicot root in many respects. It has epidermis, cortex, endodermis, pericycle, vascular bundles, pith. As compared to dicot roots, which have a fewer vascular bundles, are usually more than 6 polyarch xylem bundles in the monocot roots. Pith is large and well developed. Monocotyledonous roots do not undergo any secondary growth. A dicotyledonous stems. The transverse section of a typical young dicotyledonous stem shows that epidermis is the outermost protective layer of the stem. Covered by a thin layers of cuticle, it bears trichomes and a few stomata. Cells arranged in a multi-layer between the epidermis and the pericycle constitute the cortex. Consists of 3 subzones, and the outer hypodermis consists of a few layers of collenchymatous cells just below the epidermis, which provides a mechanical strength to a younger stem. The cortical layer below the hypodermis consists of a round with a thin wall parenchymatous cells with a conspicuous intercellular spaces. The innermost layer of the cortex is endodermis. The cells of endodermis are rich in starch grain, and the layer is also referred to as starch seeds. Pericycle is present in the inner sides of the endodermis. Above the phloem is a form of semilunar patches called sclerenchyma. In between the vascular bundles, a few layers of radially placed parenchyma tells, which consist of a medullary rays. A larger number of vascular bundles are arranged in a ring, and arrangement of vascular bundles in a characteristics of dicot stem. Each vascular bundle is conjoined open with endarc protoxylems, a large numbers of rounded parenchymatous cells with a large intercellular spaces which occupy the central portions of the stem constitutes a pith. Monocotyledonous stem. Monocotyledon has a sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by sclerenchymatous bundle sheath, a large conspicuous parenchymatous ground tissue. Vascular bundles are conjoined closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent. Water containing cavity are present within the vascular bundles. The dorsiventral or the dicotyledonous leaf. The ventral sections of the dorsiventral leaf through the lamina shows 3 main parts, namely epidermis, mesophyll, and vascular system. The epidermis, which covers both the upper surface, adaxial epidermis, lower surface, abaxial epidermis, is a leaf has a conspicuous cuticle. Abaxial epidermis generally bears more stomata than the adaxial epidermis. The later may even lack the stomata. The tissue between the upper and the lower epidermis is called the mesophyll. The mesophyll, which possess chloroplast and carry out the photosynthesis, is made up of parenchymatous. It's a two-layer cells and the palisade parenchyma and the spongy parenchyma. The adaxial placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval and the round loosely arranged spongy parenchyma situated below the palisade cells extends lower epidermis. There are numerous larger spaces and cavities between the cells. Vascular system includes vascular bundle which can be seen in the veins and midrib. The size of the vascular bundles depends upon the size of the veins. The vein vary in the thickness as in the reticulate venations or a dicot leaves. The vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. Look at the figure. Find out the position of the xylem in the vascular bundles. Isobilateral monocotyledon leaf. The anatomy of isobilateral leaf is similar to the dorsiventral leaf in many ways. It shows the following characteristic differences. In an isobilateral leaf, the stomata are present on both the surface of the epidermis and the mesophyll is not differentiated into palisade, spongy parenchyma. In grasses, certain adaxial epidermis cells along with the veins modify themselves into a large empty colorless cells. These are bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water, they are stressed and the leaves curls inwards to a minute minimized water loss. The pallor venation as a monocot is reflected in the near similar sized vascular bundle except in the main veins as in the vertical sections of the leaves. Summary anatomy. Anatomically, a plant is made up of different kinds of tissues. The plant tissues are broadly classified into meristematic, apical, lateral, intercalary, permanent, simple, and complex. Assimilations of food and its storage and transportation of water, minerals, photosynthesis, and mechanical supports are the main functions of the tissues. There are three types of tissue: epidermal, ground, and the vascular. Epidermis tissues are made up of epidermal cells, stomata, epidermal appendages. The ground tissue system is a form of main bulk of the plant. Divided into three zones: cortex, pedunculated pith. The vascular system is found to be xylem and phloem. On the basis of presence of cambium, locations of xylem and phloem, the vascular bundles are of different types. Vascular bundles are of forms of conducting tissues translocating water, minerals, and food. The monocots and the dicots and the plants shows a marked variations in the internal structure. They differ in types, numbers, location, and vascular bundles. Secondary growth occur in most of the dicotyledonous roots and the stems. Thank you for watching. You have tuned into Kalang platform, igniting curious mind where you and I grow together. Thank you. Have a wonderful